Neuroscience Chapter 4, Segment 3. Drugs may affect postsynaptic events. Effects on transmitter receptors. Selective receptor antagonists bind directly to postsynaptic receptors and block them from being activated by their neurotransmitter. Selective receptor antagonists bind to specific receptors and activate them, mimicking the natural neurotransmitter of those receptors. Effects vary depending on the particular type of receptors activated. LSD, for example, produces bizarre visual experiences through strong stimulation of a subtext of serotonin receptors found in the visual cortex. Effects on cellular processes. When they bind to their matching receptors on postsynaptic membranes, neurotransmitters can stimulate a variety of changes within the postsynaptic cell, such as activation of the second messengers, activation of genes, and the production of various proteins. Some drugs induce the postsynaptic cell to increase the receptors, changing the sensitivity of the synapses. Some drugs cause the downregulation of receptor density. Drugs that affect the brain can be divided into functional classes. Antipsychotic drugs relieve symptoms of schizophrenia. Antipsychotics are a class of drugs that alleviate schizophrenia. Neuroleptics are a class of antipsychotic drugs, traditionally dopamine receptor blockers. Examples are chlorpheniramine and hyperdrol, and loxapine. They all act as selective antagonists of dopamine receptors in the brain. They are called typical neuroleptics and are good at relieving the symptoms of schizophrenia that a dopaminergic explanation of the disease became dominant. Atypical neuroleptics feature non-dopaminergic actions, especially the blockade of certain serotonin receptors. An example would be clozapine. It's claimed to reduce additional symptoms that typical neuroleptics do not receive. Antidepressants relieve chronic mood problems. Disturbances of mood or affective disorders are among the most common psychiatric complaints. Antidepressants are a class of drugs that relieve the symptoms of depression. Monoamine oxidase, or MOA inhibitors, are enzymes that break down and inactivate monoamine transmitters. Examples are transcycloprone and isocarbazine. They break down monoamine transmitters at the axon terminals, reducing transmitter activity. This allows monoamine transmitters to accumulate at synapses, with associated improvement in mood. Tricyclic antidepressants act by increasing the synaptic content of the monoamines, norepinephrine, and serotonin. An example would be tirfenol. It blocks the reuptake of neurotransmitters into presynaptic axon terminals. Selective serotonin reuptake inhibitors alleviate depression by selectively allowing serotonin to accumulate in the synapses. Examples would be Prozac, Zoloft, or Celexa. They lack some of the undesirable effects of older drugs, but they have the side effects of their own and can take as long as 6 to 8 weeks to go into full effect. Antioxidants combat anxiety. Antioxidants belong to a category of depressants, drugs that depress or reduce nervous system activity. Alcohol and opioids are the originals, but their anxiety-fighting properties come at the cost of intoxication, additional potential, and neuropsychological impairment with long-term abuse. Barbiturate drugs are downers. They're addictive and easily overdosed. They develop to reduce anxiety, promote sleep, and prevent epileptic seizures. Benzodiazepine antagonists. They are the most heavily surprised drugs. Diazepam and lorazepam. They bind to specific sites on GABA-A receptors and enhance the activity of GABA. GABA-A are inhibitory. They help to reduce the large inhibitory postsynaptic potentials that would be caused by GABA alone. This results in the end result of reducing the excitability of neurons. They have several different binding sites, some that facilitate and some that inhibit. The benzodiazepine is a binding site is an orphan receptor, a receptor for which an endogenous ligand has not been yet identified. Allopraganol is a steroid derived from the hormone progesterone, yet acts on another site on the GABA-A receptor. It is elevated by stress and has a calming effect. It is increased by alcohol ingestion. Many other neurosteroids may act on GABA-A receptors to produce analgesic, analgesic, and anticonvulsant effects. Alcohol has several effects. The initial stimulant phase is followed by a more prolonged depressant phase. Alcohol activates the GABA-A receptor, coupled chlorine channel, increasing postsynaptic inhibition. It attributes to social disinhibition and impairment of motor coordination. Chronic alcohol abuse dangers your neurons. The cells of the superior frontal cortex, the Purkinje cells of the cerebellum, and the hippocampal pyramidal cells show particularly prominent pathological changes. Alcohol abuse by expectant mothers can cause permanent brain damage to the developing fetus, called fetal alcohol syndrome. The frontal lobes are the brain areas that are most affected by chronic alcohol use. There is a strong hereditary component to alcoholism. Periodic overconsumption of alcohol, known as binging, may cause brain damage. Damage is evident in the olfactory bulbs and the limbic structures, connected with the hippocampus, and is associated with impairments of cognitive ability. It reduces the rate of neurogenesis, the formation of new neurons in the hippocampus. Opiates help to relieve pain. Opium is an extract from poppy flower seed pods. Morphine is the major active substance in opium and is a very effective painkiller, but it has a strong potential for addiction. It's a close relative to heroin. Opioids bind to opioid receptors that are concentrated in certain regions of the brain. They are found in the limbic and hypothalamic areas of the brain and are rich in the locus coeruleus and in the periductal gray, the gray matter that surrounds the aqueduct in the brainstem. Injection into the periductal gray produces strong painkiller. This region is where the morphine acts to reduce the pain perception. Endogenous opioids, a family of peptide transmitters that have been called the body's own narcotics, are enkephalins, endorphins, and nidorphins. 3 types of opioid receptors are delta, kappa, and mu. They are all metatropic receptors. Medications such as naloxone can reverse the effects of opioid overdose. Cannabinoids have a wide array of effects. Marijuana is a dried preparation of the Cannabis sativa plant. Typically inhaled, it contains dozens of active ingredients, mainly THC. This can cause relaxation, mood alteration, which is the most common, but the stimulation, hallucination, and paranoia. Sustained use can cause addiction and accelerated cognitive decline. Frequently smoking can contribute to the respiratory disease. Adolescents who use marijuana are more likely to develop psychosis in adulthood. It's not clear whether the psychosis is caused by the drug or people who are pre-psychotic are more likely to turn to marijuana. The brain contains cannabinoid receptors that mediate the effects of compounds like THC. Cannabinoid receptors are concentrated in the stigma nigra, the hippocampus, the cerebellar cortex, and the cerebral cortex. These are 2 subtypes, CB1 and CB2. Both are G-protein coupled metatropic receptors. Only CB1 receptor is found in the nervous system. The CB2 receptors are predominant in the immune system. Endocannabinoids are endogenous ligand of cannabinoid receptors. They can function as retrograde messengers, conveying messages from the postsynaptic cell to the presynaptic cell. It's thought to modulate the release of the neurotransmitter by the presynaptic nerve terminal. The most studied is the anandamide. It has diverse functional effects, including alterations of memory formation, appetite stimulation, reduced sensitivity to pain, and protection from exotoxic brain damage. Most CB1 receptors are found in the GABAergic axon terminal. They modulate GABA release. In the case of the hippocampal neurons, this action has the result of transient amnesia, secondary to changes in protein synthesis in the postsynaptic cells. The study of endocannabinoids will aid in the search for drugs that share the beneficial effects of marijuana. Stimulants increase the activity of the nervous system. The degree of activity of the nervous system reflects a balance of excitatory and inhibitory influences. Stimulants are drugs that tip the balance towards the excitatory side. They have an altering and activating effect. Amphetamine-like stimulants, called cathinoids, are released when the African shrub khat is chewed. It's a synthetic catenoid called methadone has rapidly gained in popularity and seems to act as an amphetamine. Nicotine. Exposed to the large surfaces in the lung, nicotine from cigarettes enters the brain and the blood much more rapidly than other tobacco products. It increases heart rate, blood pressure, secretion of hydrochloric acid in the stomach, and intestinal activity. Smoking and nicotine exposure in adolescents has a lasting impact on attention and cognitive development, likely as a consequence of impairments of glutamatergic synapses in the prefrontal cortex. The nicotine chlorine receptors. Through this receptor, nicotine from tobacco exerts its effects onto the body. They drive the contraction of skeletal muscles and the activation of various visceral organs. They are found in high concentration in the brain, including the cortex. Nicotine acts directly in the ventral tegmental area to exert its rewarding/addicting effects. Cocaine. Artificially purified cocoa extract that is powerfully addictive alkaloid stimulant. At first, it was added to beverages and tonics for its stimulant qualities. It was also used as an anesthetic and an antidepressant. Crack is a smokable form of cocaine that enters the blood and the brain more rapidly. It is more addictive than the cocaine powder. Heavy use raises the risk of serious side effects like stroke, psychosis, loss of gray matter, and severe mood disturbances. Cocaine acts by blocking monoamine-transporters, especially those for dopamine, slowing reuptake of the transmitters and therefore boosting their effects. Cessation of cocaine use often produces very uncomfortable withdrawal symptoms, initial agitation, powerful drug cravings, followed by depression and an inability to enjoy anything in life. Cerebral glucose metabolism is decreased for months after use is discontinued. Users are at risk for dual dependence, which is the interaction of two or more drugs produces another addictive state. Amphetamine. The molecular structure of the synthetic psychostimulant amphetamine closely resembles that of cathepechylamine transmitters. Amphetamine and methamphetamine cause the release of these transmitters from presynaptic terminals even in the absence of action potentials. And when action potentials do reach the axon terminals, amphetamines also potentiates the subsequent release of transmitters. Once the transmitter has been released, the amphetamine further enhances activity in two ways. First, by blocking the reuptake of catecholamines in the presynaptic terminal. Second, by providing an alternative target for the enzyme that normally inactivated them. The result of the amphetamine various actions is a monoaminergic synapse becomes unnaturally potent, strongly affecting behavior. This causes an increased vigor and stamina, wakefulness, decreased appetite, and feelings of euphoria. Addiction intolerance to amphetamine and methamphetamine develops rapidly, requiring even larger doses, leading to sleeplessness, severe weight loss, and general deterioration of mental and physical conditions. Prolonged use may lead to symptoms that closely relate those of paranoid schizophrenia. Amphetamines act on the autonomic nervous system to produce high blood pressure, tremors, dizziness, sweating, rapid breathing, and nausea. If it has been chronically abused, people display symptoms of brain damage long after they quit using.